we have the current session, that is pre-lunch session of today. So proceed, Mr. Siddharth, proceed. Yeah, I
Yes,
will
sir.
start my talk in 5 minutes.
Thank you. The last session by Ms. Dipti Sharma, and the now next session is delivered by the backbone of the conference, President, Sharda Institute of Science, Technology, Education and Research, as well as Honorable Secretary of International Association of Zoologists. Currently, he also working as a assistant professor in Department of Biotechnology, MMDU, Mullana. He has a great personality. He was also working as a special officer of Wildlife Crime Control Bureau, Global Goodwill Ambassadors India, Academic Outreach Advisor Eureka US, and my guide, Dr. Sushil Kumar Upadhyay sir. Without taking more time, now I would like to invite Dr. Sushil Kumar Upadhyay sir for his blessing and kind words about the conference as well as the subtopics of biotechnology and some knowledgeable points. Please, sir.
Thank you, Mr. Siddharth. Thank you so much. I'm not actually for these words. You have even though given a lot for me. Please allow me to say so that I can proceed for presentation. So please allow me for sharing screen.
Okay, sir. Thank you, sir.
So we are here. Listen, very laureate personality, eminent speaker from various fields dealing with the advancement in biotechnology, life sciences, and ultimately we have compiled up to the wound healing, which is our thrust and focal theme of this international 3 days international event. Now, as I'm belonging to the section geology, so without dealing the conventional geology, I suppose it is not possible to lead into the modern trends into the biological sciences up to biotechnology, genetic engineering, molecular biomarkers in different aspects from its identification to its application. So, as a geologist, it's our duty also to explore the conventional geology, even though right now it is at backup path. Nobody is interested to work with conventional geology or conventional life sciences. Everybody is interested in actions, reactions, chemical sciences, biomolecular aspects, genetic engineering, etc. I hereby tried to compile the role of conventional geology and the need of advancement in this to propagate up to its extreme. That's why I have chosen these aspects, that is the biotechnological approaches in sustainability and control of parasitic diseases. As in the science, there is a reverse genetics you have been studied, and in reverse genetics, we have followed from backs out to forward. Here, from the title, I will start from the parasitic diseases. Parasites are the individual which are belonging to or any types of the categories from micro to macro individuals. Here the parasites we have discussed are the belonging to animal groups from invertebrates, basically from the phyla Helminths. Helminths as an individual which are triploblastic, acoelomate to pseudocoelomate, with diversified occurrence and ubiquitous in nature, occupying all kinds of habitats from aquatic water body to arboreal. Even though from the space, various kinds of the nematodes has been recovered, that may leads into either role in the bioremediation or sometimes it is responsible for diseases when the scientists reach to the space center. The objective of the current presentation is to explore the morphotaxonomy after the recovery of the parasites. Then you have to follow the modern statistical tool for its evaluation, analysis, and later on how it can be validated by the application of molecular genetics using certain conserved genes in these eukaryotic organisms. And after that, nothing is possible without the interaction of the environment. So when the interaction of the environment is placed with the organisms, then what kind of the problems can be faced and how it is responsible for its further modification for its better survival. In these reference, I have applied some bioproducts, that is the epitoxin, you can say that venom, and characterize its role as an anthelmintic without the other processes because it has been earlier discovered and very much roles of the V venom has been described in the biomedicines, medicinal like arthritis, multiple sclerosis, rheumatoid, scleroderma, and other diseases including the hypertension. But the anthelmintic potential was not explored. That has been explored by the School of Parasitology, University of Allahabad, formerly called Oxford of East. I was lucky to be as a student of that very school and try to find some nature of this epitoxin. It has been characterized by mass spectroscopy, but after its extraction from the live honeybee, it has been processed through the lyophilization to reduce the moisture and make it more viable, more fractionable, and more durable because mass spectro facility is not in our laboratory, so it was necessary to proceed to this for the other laboratory for that very purpose. It has been lyophilized, otherwise it will oxidize and lost its characteristics as enthalpy potential. This is the outlook how we have proceed for different processes during its analysis through MS. These are the findings out of these. The key interest is the peaks of 1.32 and 1468.7 at which the melitine as a product has been recovered with that analyzed and used as an enthalpy activity in the major constituents of the venom. After that, we have earlier also made different trainings since 2013, 2009, even though you can say that in which I have planned several trainings programs also up to the grassroots persons to make them skilled and for sustainable livelihood. During our parasitic investigation, I have worked upon large number of the fishes, even though it is under the ethical issues, but I have taken the clearance of the ethical issues from our institutions, University of Allahabad. Thereafter, I started my work on the freshwater fishes along with the marine fishes. I visited Goa as a marine coast 24 times during my PhD work for the analysis of water body as well as the parasites in last 4 years from 2008 to 2012. Meanwhile, as when I was at Gangetic region at the river Ganges, then I also collected freshwater fishes of different species, and the number you can enumerate here how much large individuals I have taken during our study. Then the hosts were brought to a laboratory, processed it for parasitic identification, isolation, and then the nematodes, cestodes, or trematodes, whatsoever the individuals were obtained, processed by 4% hot formaldehyde, lukewarm water, as well as glyceraldehyde for its further morphotaxonomy. Some of them has been placed for pepsin digestion to assess the stages from its tissues. Some of them has been placed into molecular grade alcohols for its molecular analysis. Some of them has been placed into 6-NaCl for its biochemical assay. All these aspects we proceed as a conventional research ethics. That's why we are able to go for its correct finding. I am just sharing these things for the young ones which are attending this conference, because when they are moving in research area, they feel several kinds of constraints how we proceed ahead. Therefore, I have given here one by one step. Like a PhD student, just you have to follow these steps, then you can find your results without any kinds of constraints within stipulated time. Further, for the molecular analysis, there is a need of the bioinformatics. It is only possible when you have to the skill of bioinformatics, so that the sequences you can obtain can be blasted, analyzed, compared by using several modern tools like Mega, ClustalW, PhylogenEFR for its correct interpretation. What are the need of the parasites to investigate them? As far as the individual is concerned, it is the need for biodiversity conservation. As far as its activity is concerned, it is responsible for several diseases in all kinds of individuals, from Pisces to human beings, even though these are also found in several arthropods. Therefore, it is urgent need whenever these kind of fishes can be eaten by human beings, it may possible it will reach to the higher trophic levels and may cause lethality into the human beings also. But all parasites are not able to do such activity. Only few of them are able to cause damage into the human beings when it reaches through the food substances, edible substances. It may be grain in which the eggs are present that can be used by the human beings. Sometimes when the pupils depends on the aquaculture products, then they may also got damaged, infected by these kind of parasites by cooking uncooked, undercooked or semi-cooked foods. And then these parasites may lead to the human beings, take the position into the gut muscles and causes the major damage up to the liver cirrhosis, even though echinococciasis may lead into the cancer, as was discussed by chief guest of this event, Professor Sandeep Malhotra sir. I was enough lucky to have this kind of supervisor. I have worked as a student, PhD student with him also. This is the schematic presentation. You can find it in any book, but I have correlated my findings with this ideal picture. This is the study Rima multiembryonata, has been published in 2017 in Species, a good impact journal, and thereafter these are the eggs and stages from the host and further its modification for its life cycle. Therefore, I have tried to compile it in correlation with this standard I presented here. It's a hypothesis. It is still in the process through in vitro studies. The in vitro studies were also performed in the experimental model, and the model organism was fed by the infected embryonated eggs, and further its symptomatic condition being recorded, and thereafter the histopathological sections may also confirm its occurrence into the liver region. After its confirmation, it has been assessed through the gut parasites, and this adult individual has been recorded. Therefore, its genetic potential can be proved by this kind of experiment during our PhD study. Then how the parasites it looks like. You cannot imagine the parasites may be in such a good architecture, having such a good architectures. This is Sturdinema multiembryonata. Whole body is spiny with 3 lips. These are the primary characteristics of the nematodes, and you can record it. But those who are dealing with these parasites are very much aware. This is the scanning electron microscopy, and you can find the demarcated feature of the male and female individual. This is the spicule of the male individual. The populated distributions, when is further magnified through the scanning electron microscopy, this kind of good architecture you can record it. You can record it that the populated structure were further surrounded by several spines-like structure, which helps in the grasping of female during its copulation. This is the scanning electron microscopy was captured from All India Institute of Medical Sciences, New Delhi. This is the handmade diagram of these individuals called camera-Sharda diagram. I have prepared, this diagram was prepared by me during 2010. Further molecular validation was done after finding the genomic DNA processed for PCR. After PCR amplification, the gene was sequenced by the kind courtesy of Microgen Industries, Korea, and the raw sequences obtained were blasted by using Mega. Further, the necessary editing was done by the application of bioedit, and then by the application of cluster W and phylogeny, or this kind of never joining dendrogram was created. And in this, you have been find it. This is the comparative account of the Sturdinema, and later on, it was showing its demarcated phylogeny from the other existing parasites of this category. This was the Sturdinema showing other. I created a subfamily among the Spiroradians, which also has been published in 2016. This is another diagrammatic illustration with maximum likelihood. If all the characters means all the illustrations going similar characters, then you can belonging word. Otherwise, if any ambiguity is being recorded into the phylogenetic dendrogram, then you have to assess it further for its validation. This is also an illustration of K2 parameters, evolutionary relationship of the Spiroradians. This is another example of the parasites, that is anisakis. I am just showing a very good diagram of the parasites, because whenever you just listen about the parasites, individuals trying to far away, ये बहुत गंदा होगा, कैसे होगा, पता नहीं कैसे दिखता होगा, लेकिन you can see here the beauty of parasites. Parasites are not always ugly. These are very beautiful than me. I'm not so beautiful like these parasites. You can assess how much quick mouth opening of these parasites. This is the snake-like structure, but it is a diacline parasites. These are the anisakis. All these are having the genetic potential. So those which are feeding on fishes, I wish to request him before feeding, you have to analyze that either they having parasites or not. Otherwise, you have to overcook the flesh and then eat. Undercooked and semi-cooked foods are not allowed to eat. May possible you have been infected by these kind of parasites. As I'm saying here, semi-cooked or undercooked, people believe that overcooked food, where is the possibility to get infected. But if you move across the coastal areas, you can find the people directly eat the flesh of several fauna as an interest of the diet. Then, and it has been recorded that the coastline areas have been very much infection of these kind of parasites. Further, you can assess how much beautiful of the parasites are. This is the slide prepared by me during my earlier course of the study. You can find the internal structures even though this is the स्कॉलेक्स ऑफ द टीनिड वर्म टीनियोसोलियम का नाम आपने सुना होगा अभी तक, लेकिन आप यहाँ देख सकते हैं कि हाउ मच इज दिस. You are looking these flower-like structures like a flower of plants, but it is not a plant. It is a parasites belonging to the cestode category. This is the scolex region with 4 bothrial apparatus and that giving an appearance of flower and the name belonging to its the tetraphyllid scolex. This is the parasites which is belonging to the hardheaded category. This is the scolex of these. It has been recorded from गज सूज. आप सोरेक्स का नाम सुने होंगे, ठीक, छछूंदर जिसे हिंदी में कहते हैं अपनी लोकल भाषा में. तो ये छछूंदर से निकाला हुआ मेरे द्वारा एक पैरासाइट है. This is the mature proglottids. This is the mature and gravid proglottid of this tetraphyllid worm. This is the stenid worm recovered from the gut of model mice. You can also assess the beauty of these cestodes through the scanning electron microscopy. आज तक आपने बहुत से छत वाले मंदिर देखे होंगे जिसमें ढेर सारे गुंबद होते हैं, उनके ऊपर एक क्राउन होता है, लेकिन these parasites also have very good crown overhead. These are the sucker-like structures. You can ground parasites here. This is the Gangesia. This is the Hymenolipis. This is the Senga. This is the Polyoncobothrium. This is the Sorex. And you have been find all kinds of these beautiful parasites, and all are recovered from various sources belonging to Pisces to mammalians during our earlier finding. Still, I am working on these lines to find this enigmatic ultratropography. You have been recorded that how the nanoparticles are adhered over that. These are the some interactions studied, and I wish not to explore many more because I was studying about the entelmethic potential of some interesting chemicals, and I got found how the damage in the cuticular lining of these parasites can be recorded. These are the major findings. I'll try to find the more accurate aspects, and thereafter, whenever may possible, I will describe in detail. These are the morphotaxometric findings. And then now I'm coming to the next type of parasites, the trematodes. I just earlier described nematode and cestode. This is the trematode. Cephalogonimus semini has been published at first in 2012. I have published its 4-volume, 4 parts in different high reputed journals since 2012 to 2018 continuously. This is the trematode. You cannot imagine how much these golden particles are present inside it. You can collect these golden points from that and become as a billionaire. But actually these are the eggs. These are the eggs present inside these structures of the hermaphroditic individuals, because both kind of structures, male and female structures, are present in these. So you can find the beauty of these cephalogonimus. I am showing only one individual here. This is the handmade diagram through the camera Lucidia by me, showing all structures of the cephalogonimus. Has been published in Jutaxa. This is the comparative account. And then the environmental interactions, you can find it when I was working with the lab, parasitology lab in University of Allahabad, how I was. I just become old with these white and black beards right now. But at that moment, I was visiting these structures. You can find the sites. This is the loving structures. You can find it. This is the also structure of estuarine area near Hooghly River, where the Gangetic region is connected with the Padma River in Bengal. It is, I suppose, nearly 45 kilometers away from Digha. Digha का नाम आपने सुना होगा, Midnapur, Digha. All are the areas. I have wandered a lot and spent many more months there in that very areas. These are the coastal lines of the Goa region. You can find this kind of ferry here is giving its appearance. This is the laboratory in which I was working, parasitology laboratory. Parasite is, all things are available here, and after working, I am just drinking a sip of the tea. Without any kinds of hesitation, I was working with the parasites at the same time and drinking tea by other hands. These were the sites of the market of the Goa when I was collecting the parasites. From the sites, you can find it. These are the structures. I suppose you are very much aware about this. This is the transmission electron microscope. This is the gold coating. जब आप किसी भी इलेक्ट्रॉन माइक्रोस्कोपी के लिए जाते हैं तो either you have processed by carbon coating or gold coating. This is the instrument for the gold coating. This is the river Ganga at Allahabad. I was collecting the sample. This is the GPS for recording the site and local, means latitude, longitude. I was inserted this slide for a breathing period because when you are presenting your slide continuously through the scientific matters, students may not prefer to listen always science. Therefore, when you wish to prepare any presentation, you have to insert some beautiful pics inside it to be relaxed at that moment. That is called breathing slides. This is the oscillations in the hydrobiological parameters of marine water body has been assessed through the time series plots. And as we know that as the temperature rise, dissolved oxygen concentration is reduced. You can record it here through the illustrations. This is the minimum burden as well as the Poisson correlation of the parasites. It is stimulated and analyzed manually. I have calculated by using calculator manually, not using any software. This is the principal component analysis. After that, not easy to conclude the findings, but still I tried to conclude the findings. The antibacterial and anthelmintic potential of epitoxin in vivo, in vitro, both by using several assay techniques. The statistical application reflected that constituents of V venom, that is heptapeptides, play a significant key role as an antibacterial and anthelmintic potential. Therefore, the conservation of these natural resources must be applied for biotechnological application. Molecular characterization tool reduces the ambiguity in narcotaxonomy by conventional activities. Physicochemical factors may also regulate the host specificity of these worms. Therefore, it is always advised don't disturb the water quality and natural resources in which these flora and fauna are residing that may cause damage to the human beings by passing through these aquaculture feed substances. These are the bodies which are from which I have received several kinds of fellowships from JRF, SRF, postdoctoral fellowships. So thank you so much for patience listening. This is our current beautiful campus, MMEC. I suppose most of the students among you are very much affectionate to this beautiful campus. So whenever you have time, please visit at once this MMDU campus, Mullana, Ambala. So thank you all for patience listening. If you have any query, please, I wish to clear all the aspects of these parasites, biostat, taxonomy, biochemical analysis, etc. So most welcome.
Thanks, sir, to deliver a very good and very informative lecture.
Yes, sir. Thank you so much, sir.
Very nice, sir. Very nice. And you are also old days and now also.
Yes, sir.
Thank you, sir. So now we have a short break of lunch. Thereafter, we have very beautiful session of young students as young scientist presentation. We have very good number of the young scientist presenters in our hand right now. There are 25 contestants for oral
Okay, very
presentation
good.
and poster presentation. So I wish to request you all, please attend that very session to raise the status of the students, because they always try to place their findings at a certain place. So we have very good and very intelligent students with us. They will present their presentation either oral way or poster way. After that, our jury members will analyze their poster, and among these, I assured that it will not be possible to assess the best one, but still I request to our jury member to find the best one, posters and oral presentation, and best three will be awarded by the merit of excellence along with some cash prize money. Even though it is not possible to me give the cash in hand right now, I will send them the check or through the Google Pay, Phone Pay, etc. in their personal account. So I wish request to you all, please be available at, I suppose, 2 p.m. for listening to these young scientists and their moral boasting also. So thank you so
so
much,
much, sir.
Dr. Manoj Singh sir, if you wish to say.
Thank you so much, sir.
Sushil, thank you for your wonderful talk. We must appreciate Dr. Sushil for conducting this international virtual conference on seminar and asking the student also to participate at their end and to felicitate them or to they make them happy. You are giving some prizes in terms of some cash prizes. That is a big achievement for them, I should say. And I hope they will participate. They will give their talk at the best, and we'll see in the next session. Thank you so much.
Yeah, thank you, sir. Thank you, sir.